Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump in today, if you like what we do, please remember to hit like and subscribe. It really helps support the Science Experience. Okay, so today we're embarking on quite a deep dive. We're looking at a really interesting new paper from Nature Methods, and it tackles a pretty fundamental challenge in scientific discovery, right? How do you quickly and accurately figure out what groups of genes actually do?
Absolutely. For researchers, making sense of these huge data sets, especially gene data, it's just crucial. You need it to uncover biological mechanisms, disease pathways. But as we've discussed before, large language models, LLMs, they can struggle. They often have problems with factual accuracy.
Right, the hallucination issue.
Exactly. They can confidently sort of invent information. And in science, that's not just a small bug, it's a major roadblock. So our mission today is to unpack what looks like a genuinely groundbreaking solution to this, specifically for genetics.
Okay, let's get into it. The challenge is clear LLMs can kind of go off script. So what's the innovation here? What's the architecture?
It's called GeneAgent, and it's an AI agent built on GPT-4, designed specifically for analyzing gene sets.
GeneAgent, okay.
And its most distinctive feature, the real clever part, is this powerful self-verification process. It essentially makes it self-correcting.
Self-correcting. How does that actually work? How does it minimize those hallucinations you mentioned?
Well, what's fascinating is how it does it. You can think of it like an AI researcher that just constantly needs to double-check everything. It autonomously interacts with, I think it's 18 expert-curated biological databases, uses 4 different web APIs.
18 databases, wow.
Yeah, to rigorously fact-check its own output. So it's not just generating an answer and stopping. It actively questions itself. It's almost like it's thinking critically, more like a human researcher would. It's a big step for AI trustworthiness, really.
There's a process to it.
A 4-stage pipeline. First is generation. It makes an initial hypothesis about the gene set's function. Then, the key part, self-verification. That's where it hits those databases and checks its claims.
Okay.
If it finds problems, discrepancies, then modification happens. It revises its output. And finally, summarization. It presents a refined, validated, and hopefully trustworthy description.
That self-verification stage sounds like the absolute core of it.
Yeah.
But, how much better does it actually perform? Do we have numbers comparing it to just, say, standard GPT-4?
We do, and the evidence is pretty compelling. GeneAgent consistently outperforms standard GPT-4 in these tasks. For instance, they evaluated it across over 1,100 gene sets, 1,106 to be exact.
Right.
And GeneAgent showed significantly higher semantic similarity scores.
Okay, semantic similarity. What does that actually mean in practice for analyzing genes? Is it just getting closer to the right answer?
Pretty much, yeah. It means its descriptions of gene functions were much closer in meaning and context to the known biological truth. To put a number on it, GeneAgent got over 90% similarity to the ground truth, the established functional name, in 170 cases. GPT-4 only managed that in 104 cases.
That's quite a jump.
It is. And about that self-verification reliability you asked about, manual review confirmed it was correct 92% of the time.
92%. In biology, where details matter so much, is that accuracy level really a game changer for this kind of AI?
Oh, absolutely. That 92% means the system is largely dependable on its own. It significantly cuts down the need for a human to constantly check its homework,
Yeah.
It frees researchers up to do the actual research, the deeper analysis, the discovery part.
That makes sense. So it works well on benchmarks, powerful internal logic. But what about, the real world? Did they test it in a practical scientific setting?
Yes, and that's the crucial test, isn't it? They applied GeneAgent to 7 novel gene sets. These came from mouse melanoma cell lines, which is a really important area for cancer research.
Okay, so new territory for the AI.
Exactly. And expert reviewers confirmed it. GeneAgent produced functional descriptions that were more relevant, more comprehensive than GPT-4 could alone. It even turned up some potentially novel biological insights that weren't immediately obvious. Plus, it proved robust across different species, which is also important.
So it's adaptable too.
Right. It really seems to merge the strengths of LLMs, their ability to understand and generate complex language, with traditional, rigorous methods like gene set enrichment analysis. So it's not just about getting results faster. It's about combining that AI explanatory power with the solid, vetted knowledge we already have. It gives you results and clear explanations for why they matter.
So let's zoom out. What does this all mean for you, the listener, and maybe for the future of science research more broadly? It seems GeneAgent is a pretty significant leap. It tackles that AI hallucination problem head on by basically building in a fact-checking mechanism using reliable, curated knowledge.
Exactly. It's like giving the LLM its own internal scientific editor, constantly making sure it sticks to the verifiable facts.
That's a great analogy.
And think about the wider implications for you. If AI can learn to verify its own scientific claims using external established data, well, what other fields could use a system like that? It's a really powerful model, I think, for building more trustworthy AI systems everywhere. It moves us closer to AI being not just a generator, but a reliable partner in discovery.
Fascinating prospect indeed. More trustworthy AI across the board. Thank you for joining us on this deep dive today, and please remember to like this video and subscribe to the SANS Science Experience for more deep dives into the cutting edge of science.